This is SciFi Talks. Today in this episode, we would like to welcome Dr. Sunil Acharya. Acharya is an alumnus of the MD Anderson Cancer Center UTHealth Houston Graduate School of Biomedical Sciences. He currently holds the position of Principal Research Scientist at Dr. Katy Rezvani's laboratory at MD Anderson Cancer Center. He is focused on harnessing the potential of NK cells as therapeutic agents. Let's welcome Dr. Acharya.
Welcome, Dai.
Thank you.
I would like to start from your journey. So how was your journey from Nepal to one of the leading lab in the cell therapy field?
Yeah, first of all, thank you, Sudarshan, for this opportunity. Okay, I really loved what you're doing, and thank you for. I'm very grateful for this opportunity. So, back in Nepal, when I graduated from high school, I was looking for the colleges, and at the time, the biotech was very, very popular and very booming. There were a lot of biotech industries, and biotech was just booming in US and in India, but in Nepal, because that in Nepal, like the Kathmandu University, they recently opened a biotech undergrad program. So it was very popular and very exciting, and someone was interested in science. That led me to join that program in Kathmandu University, and we finished it. But the unfortunate or fortunate, I don't know what to say, in being in Nepal, there is not much job opportunities in biotech that right? At that time, I'm talking about in 2007, 2008. I know now people are doing great things there too, but at the time, so the only option for us at that time was to go out and get higher education. So that led me to come to US to pursue my master's. And after that, when I was working in one of the small city called Tyler, and I was doing the master's there, and it was University of Texas Health Science Center at Tyler. And when I was there, we used to hear about this very amazing cancer hospital called MD Anderson Cancer Hospital, which is also part of University of Texas. So that was always fascinating. So what I did was, after my master's, I applied for a PhD program to MD Anderson because they have this graduate school too, because I was always interested in cancer, to study about cancer. So I got enrollment in graduate school in MD Anderson, and I started my graduate school, and that's the story. After that, it's been now almost 15 years. I have not left MD Anderson. I did my PhD in MD Anderson, and when I was about to graduate, this cell therapy was picking up. There were a lot of CAR T therapies coming up, and also and just at 2000, I graduated in 2018, and 2017, the first CAR T therapy was approved. So the field was very hot. So that led me to apply to this lab, which was very immersing lab at MD Anderson. But instead of working on CAR T field, the lab was working on the CAR NK field, which is the another platform for cell therapy options. So that led me to this Katy Rezvani's lab as a postdoc, and I decided to stay there. And now I'm a principal research scientist in the lab. That's my journey.
So what attracted you to the NK cell therapy? There was T cell therapy, which was advancing. I think there was already the picking up. There were already clinical trials, many more going on. But what attracted you to the NK?
Yeah, so that's a great question. So as I said, when I was about to graduate, if I was a graduate student and if I was doing a CAR T therapy, it would have been great, right? But I didn't understand that when I started the graduate school. I went into this cancer field. I was studying this breast cancer metastasis, because at that time metastasis was so popular. But now, when I was about to graduate, CAR T field, the CAR T therapy was already approved, right? It was just approved, and there's a lot of competition, and we already know CAR T works, right? But even though CAR T therapy is great, it comes with certain drawbacks, right? And one of the few very drawbacks of CAR T therapy at that time was cost, because it was very costly. And along with this cost, it also has a lot of like toxicity, right? CRS, like cytokine release syndrome and stuff like that. So I was going to this grant rounds, in MD Anderson. It's a huge in hospital, like we have like 27,000 employee. And as a graduate student, I was looking for postdoc labs and what field to explore. And I went to this grant round. And in that grant round, Katy was giving a talk, Dr. Rezvani she was giving a talk, and she was talking about this CAR-NK therapy, and she was treating patients with CD19 CAR-NK field, and that paper is published in New England Journal of Medicine in 2020, and she was talking how this alternative source, which is NK cells, when you express a CAR on those small NK cells and give it to patients, one, they don't have toxicity. And second, it is allogenic source. That means from one source, you can develop therapy for multiple human beings, and you can make truly off-the-shelf product, right? Because so far, all the CAR T cells, even at that time, at 2017-18, when there were only 2 products, and now coming back to 2025, where we have 6 products, all of these products are like autologous product. That means the patient T cells is extracted from patients, and then we make CAR T cells based on that product, and that medicine can be given to only that one particular patient, right? But here you have another source where you can choose a healthy donor and make multiple vials of drugs, and you can freeze them and use it as a truly off-the-shelf product. So those 2 things like caught my attention, and I really loved it and thought that this would be an amazing field to pursue, and that's why I was fascinated and joined her lab as a postdoc.
Okay, so we are already in the NK cell therapy, but before that, I would like to know more about I would like to know briefly about the cell therapy. How do you define the cell therapy in layman language, or the cell therapy and NK cell therapy? Like briefly, how do you define these terms?
Okay. Yeah, the term itself is very explanatory, right? Cell therapy it's a therapy where you take cells, like it can be any cells from the human being, or sometimes cell lines also, like because there is a NK92 cell line, which is also used in the cell therapy field, and use it as a cell therapy, right?
Yeah.
So that's the name itself is like it's very explanatory, and it's not the new field. It's been there for many years, right? Like to be honest, even a part of vaccine is also originated in early years because of cells, right? They used to take the cells and they used to attenuate these cells and then make these antigens and use it as a virus, right? It's in early days. So, but in stem, like first, it was very popular as a stem cell therapy, right? Because for those people whose bone marrow was not working, or those people are doing this stem cell therapy for many years. So with those very long history of understanding of how to use these cells as a therapy, and then people also started, when this cancer was booming, and people were thinking that this cancer is a disease where our own immune cells failed to kill those cancer cells or attack these cancer cells, right? So that's what the scientists were thinking. How can we make our own cells to fight against this cancer or fight against this disease? So that's how the cell therapy field emerges. It got very successful in stem cell transplant field, which people are still doing it, bone marrow transplant or other kinds of transplant. And for T cell field, what happened is, before like this CAR T cell became popular, you had to understand there was this immune checkpoint blockade therapy, which actually give this boost to this CAR T cell therapy field, because where the doctors like Jim Allison from MD Anderson, who made this CTLA4 antibody, which binds against to the CTLA4, which acts as a brake for these T cells, because when people has a cancer, their T cells are dysfunctional, and then scientists found out that the markers like PD-L1 or CTLA4 they are expressed in order for these T cells to be dysfunctional. So they designed this antibody against this marker, which led to revolutionary revolutionize this field of immune cell therapy, right? And which leads to this now very sophisticated CAR T cell therapy, macrophages is coming up, NK cells. It's just a way of choosing the cell of your interest and then engineering them in order to make them as a source of therapy. It's same like using small molecule inhibitors or any other kinds of therapy. It's just you use the live cells as a therapy. That's why it's called cell therapy.
You said that the NK cell has one of the best feature, the feature of the self nature, right? But so is that the only feature that is making NK cells the best alternative for the T cell therapy, or are there other benefits of using the NK cell therapy in terms of infiltration or other challenges that are in the T cells?
Oh, that's a great question, Sudarshan. I wish I had a complete answer for that, but we still need to do research on it. But let me go back a little bit. So I know I said NK is suitable because you can use it as off-the-shelf product, but along with that, you need to keep in mind NK cell by itself is a natural killer cells, right? They are built to kill the either virally infected cells or cancer cells even before T cells act upon those cells, right? It's a part of the innate immunity. So whenever you get infection or virally infected, our NK cells will go and attack these cells and in order to make us healthy, right? So unlike T cells, NK cells has the innate property to kill these infected or cancer cells. So that's also one of the advantages of NK cells, because in T cells, in order for T cells to kill these cancer cells or infected cells, they need to go and recognize. It can only kill based on either binding to the antigen, which is expressed on HLA molecules, or these CAR should go and bind to the antigen, right? So T cells can only kill by this CAR specific manner, but NK cells, being a natural killer cells, they have this innate receptors, activating receptors, inhibitory receptors, so they can kill these infected cells with other mechanisms too, not only by this CAR specific manner. So that's also an advantage, right? But saying that, and also being that it can be used as an allogenic source because it doesn't have this graft versus host disease. It doesn't cause graft versus host disease like T cells. So those two are very major advantages. But when it comes to other things, we are still trying to understand. Yeah, of course, NK cells have a different properties in order to home into these different organs. So maybe we can make these NK cells home into this tumor bed or to these infected sites more effectively. So as I said, the CAR NK cells has innate ability to kill, right? And then we can use it off the cell. But regarding this persistence and homing, I don't think we can say that NK cells are better than T cells at this point, because there are more research need to be done and understand how we can make these NK cells persist longer or home into these organs. So
I would also like to know about how the engineering of the NK cell, how you engineer the NK cells. Is it that the process similar to the T cell engineering, or is it completely different than the T cell engineering?
So it is slightly different in terms of which vector you use to express these CARs into these cells. So T cells, so far they usually use this lentiviral vector, but what we found in our lab, or now many people has validated, is somehow NK cell doesn't like lentivirals that much. There are certain kind of lentiviral, I think some people are using it, but in our hands, retroviral works very good in terms of CAR transduction into these NK cells. So instead of, unlike lentiviral, we use retroviral to engineer the NK cells. That's engineering, right? Engineering, and sometimes people also use different co-stim domain in terms of NK cells or T cell activation. But what we found, and actually that's one of the postdoc work that I published last year in Cancer Discovery, is we can use CD28 co-stim, which is a T cell centric co-stim, and it seems to be work fine in NK cells as compared to other co-stims. So that people were thinking maybe NK specific co-stim is better compared to T cell co-stim, but we found out that even T cell centric co-stims, which is CD28, works better, fine in NK cells. So that's what we found. But in terms of cell expansion and cell CAR-T production, it is slightly different because T cells, they don't need any feeder cells, right? You can just stimulate them with CD3 or CD28 beads. But for NK cells, so far we use feeder cells in order to expand these NK cells, and it takes like a week for these cells to expand. So there is slight difference in CAR construct based on this vector difference. And then there is a slight difference in production when you compare to T cells when you are trying to make this NK product in GMP setting.
So in terms of handling or doing the experiment in NK cells, how challenging is it? Like in T cells, I think most of the protocols are already optimized, and so maybe a little bit easier. But in NK cells, what kind of challenges do you see in terms of handling and the normal experiments you do in day-to-day experiments in the lab?
Yes. Yeah, that's a great question. Of course, we had to spend our lab. We spent first 5 years just to optimize the CAR transduction. People will not realize that because people will only see now that we are able to make this CAR product. But what people don't realize is even before I joined the lab, our lab was optimizing this CAR transduction. We tried everything: lentiviral, baboon, retroviral, and we found out that retroviral vector was working. And then the transduction conditions, which day we need to transduce these NK cells. So there are a lot of research and time and effort has gone behind this optimization of NK cells, and now we are in the space or where we are so confident in our CAR NK transduction and manufacturing that we can transduce our NK cells with 90% of CAR positivity. We're that confident, but we spend 5-6 years just to optimize that. So there is difficulty on that, but once you optimize those things, then it's easier. Right now, we are very confident on making this CAR NK percentage. It's up to us how much we wanted to make this CAR NK positive on this CAR expression. Just to be on a safer side, for clinical product, we tend to reach between 50 to 60% CAR transduction. But if we want to make them 100% transduced, we can do that. We can easily do that. And also what kind of cytokines to add, and we also did a lot of research on these feeder cells, which kind of feeder cells to use. So these are a lot of things we have optimized. Not only that, keep in mind, unlike T cells, which is auto product, right? So the only source is you are using the patient's T cells. But for NK cells, since it's allogeneic product, there's different sources of NK cells, right? For example, our lab, we only focus on core blood derived NK cells. It's because of the feasibility, and we have access to core blood bank. So that's why we use it. But there are other sources too. Some people use iPSC derived NK cells. Some people use peripheral blood derived NK cells. Some people use NK cell lines. So if you use different source of NK cells, then again, you have to spend some time in optimizing these processes, because the process by which we are getting 100% transduction on our core blood derived NK cells, the same process may not be true for either iPSC derived NK cells or peripheral blood derived NK cells. So yeah, there are certain challenges, but we have now optimized, at least in core blood derived NK cells, the transduction, production, and all those things in the lab.
So you also said that one of the challenges of NK cell could be the homing, right? When we compare with the T cells. So how do we deliver the NK cells to the right tumor? Is it localized delivery is the right choice or the best choice, or can we do the systemic delivery too?
Yeah. So let me correct you a little bit there, Sudarshan, because I think even for T cells, homing is an issue at this point. Okay, it's not that T cell, because you see all the FDA approved CAR T cells are only for liquid cancer. There is no CAR T so far approved for solid cancer, right? Because so for solid cancer, we are up to the same speed as CAR T cell, right? CAR NK. But saying that, yeah, of course, homing will always be an issue for solid cancer. That's one of the biggest challenges that we face in order to treat solid cancer, right? So for what people are doing and what we are also trying to do is, like, like NK cells has this tissue resident phenotype. So your organs has NK cells already within it. So we are trying to understand what are these receptors on these cells which make them reside into these different organs, right? So what I believe is we need to, when we're trying to make this CAR NK product, it has to be organ specific or disease specific. That's this is completely my hypothesis. I might be wrong too, but we need to express those receptors which allow these NK cells to home into this organ. For example, if we are talking about the cancer cells in liver, or we are talking about the cancer in pancreas, maybe the homing receptor might be slightly different. So we might need to engineer them slightly differently to express these homing receptors in order to home into the organ. So that's my thinking, that's our lab's thinking, and that's what we are working on. And but so far, this is still a work in progress. A lot of labs are invested in it, and we are invested in it too. So whoever can figure it out and whoever can make these NK cells or T cells home into the tumor bed and kill these tumors, they will win the race in terms of solid cancer treatment, right? So that's the thing. But so far, I think and also the other, sorry, I forgot to mention that it also depends on which disease you are treating, right? For some disease, you cannot access the solid cancer, so you have to use the systemic delivery, right? Because if you cannot access the solid tumor, but maybe in future, in some solid cancer setting, maybe you can use localized delivery. For example, we are doing this trial on GBM with our NK cells, where we are giving the NK cells directly into the tumor. So because NK cells cannot, if you give systemic delivery to this GBM, they may not reach to the brain because of the blood-brain barrier or some other sources. But when surgeons are going and taking out this tumor, they put the Ommaya reservoir in the brain, which allows us to give our NK cells directly into the tumor. So there is a lot of research going on. So in future, I think it might be tumor specific. For example, for certain kind of tumor, we can do local delivery, which are accessible, and for certain kind of tumor, we had to do systemic. And for the systemic thing, we need to engineer our NK cells certainly so that they have these homing receptors which can help them to home into these different tumors. So, but there is a lot of research still to be done.
Anyways, are we targeting the same targets like we are targeting to CAR T cells, or the targets are completely different in terms of NK cells?
So no, we are doing the same targets for I think the benefit of NK cells, at least for liquid cancer, right? So CD19 is that's the one you'll not find any other marker like CD19. It's like it's a luck that CD19 was found as an antigen for these lymphomas, because it's widely expressed and it's only expressed mostly expressed in cancer cells and accessible, so you can target it. But when you come to solid cancer, that's not the case, right? The first thing, you won't be getting this universal expression of the antigen, right? Because there is so much tumor heterogeneity in solid cancers. And then even if you found some universally expressed antigen, for example, HER2 in breast cancer, some of the best cancer, but when you treat these cancers, somehow those cancer will develop some defense mechanism, or there might be tumor cells that can evolve which are HER2 negative, right? So when it comes to solid cancer, it's a little complicated, like in CD19 in case of lymphoma, where CD19 is universally expressed and stuff like that. So, but in terms of target, yes, we are using the same target like how T cell field is using. It's only that maybe we'll be using different co-stim, or we need to add IL-15 construct, like IL-15 construct in the CAR, in the CAR construct, in order to make our NK cell persist longer. But in terms of antigen extracellular domain, I think we can use same thing in NK cells as well as in T cells.
Are there also clinical trials in the NK cells in the liquid tumors or the lymphoma? Are there?
Oh yeah, there are a lot of NK trials, not only in liquid tumor, even in solid cancers also. There is so CD19, as I said, we initiated with the CD19 CAR NK therapy, and you can name it whatever antigens there are. I think there is some CAR NK therapy for those tumors, both for solid as well as liquid. I can talk about our lab. Our lab has so far now 12 clinical trials running, 4 or 5 for liquid cancer, like for AML, for lymphoma, and we also have most of them are for solid cancer, like pancreatic cancer or ovarian cancer, GBM, and stuff like that. So there are a lot of clinical trial ongoing in NK therapy, and there are a lot of small biotechs and big companies are also interested in NK cells, and they're doing a lot of clinical trials.
So how do you define the status of clinical trials of NK cell therapy, and what do you think, where are we failing, so in terms of translating the NK cell therapy in the clinical trial?
Oh, that's $1 million question. So yeah, we are there are a lot of clinical trials, but the sad thing is, so far there is no FDA approved therapy in terms of CAR NK therapy, right? I think the thing is, CAR T field is way ahead because it started so early, and they had a very long head start in terms of liquid cancer, and they saw remarkable efficacy, right? So that bar is super high for NK cells to meet. Right. I know we have less. The cost is less, but at the end of the day, the efficacy is what it matters. I think, right? I cannot say that because I'm working in NK cell, but it's going to be a little ironic when I'm saying this. But NK cells, the efficacy is not there yet, or we are not. All these trials are not able to show the same level of efficacy that CAR T showed in lymphoma trials by this CAR NK therapy, right? So even though the cost is less, cost can be less, or even though there is less side effects, the efficacy is what it matters. I think that's what it's a drawback for NK cells. And now there is so much research going on to manage this toxicity, and that's why the toxicity is also a lesser of extent for CAR T field, right? Because now there is a lot of drugs, and then clinicians also learned a lot, so they can manage those toxicity pretty well for CAR T therapies. But saying that, CAR NK and CAR T therapy hasn't been approved for solid cancer, so there is still some space for CAR NK therapy in terms of getting it approval in solid cancers, but hopefully in near future there will be some CAR NK therapy that will be approved in hematologic malignancies too. So I think that's our goal. Maybe we'll be the first one to have a clear FDA approved product, but let's hope that in near future that will be a case.
Sometimes I think that in the search of alternative to T cells, we are maybe targeting the different cells, like NK cell is the one, and the macrophage also. But ultimately, I think it's the combinatorial therapy, like how we take a benefit of all these kind of cells to get a synchronized benefit, right?
Yeah, that's definitely that's going to be a future, because even when we are doing these clinical trials, we quickly learn that our NK cells they interact with the host immune cells like T cells or monocytes. So definitely and even though you keep in mind, even CAR T therapy has shown a remarkable efficacy, it's not curable for all patients, right? There is curable for certain percent of patients, but large percent of patients are still relapsing. The therapy only lasts for a couple of months, right? So still, definitely, that's, I don't think CAR T or CAR NK, there is it's going to be a magic bullet. So we definitely need to combine it with either other cell therapies or other chemotherapy or small inhibitor molecules or any other kind of therapy. That will definitely be a future. And also personalized therapy, some vaccines maybe, like now there people are working with using vaccine therapy for the immune therapy too. So definitely that will be the future. And depending on the tumor types and then where the patient is, especially like it's a very good context, like as we both know, we are from Nepal, a very like a country with not as rich as US, right? Like third world country. I think NK therapy might be very suitable for these kind of countries where people cannot afford these super expensive CAR T therapies and other kind of things, right? And the thing is, and India is doing great. I don't know that or not, but India now has CAR T therapy, which is 10 times lower the cost of CAR T therapy here in US, right? So there is a lot of room for improvement. There is a lot of opportunities for all these kind of therapies for the patients, and it's for the betterment of the patients, right? So let's be hopeful.
Yeah, and also maybe this kind of cell therapy, maybe the technology is the same. Maybe if we can use the same technology in other diseases than just cancer, maybe in other diseases, maybe NK cell therapy or macrophage therapy could be more relevant, right?
Yeah, of course. People are actually NK cell therapy. It started with a transplant, not as a CAR NK, and people are still using it. And also now people are using NK, both CAR NK and then CAR T therapy in terms of autoimmunity, which is a huge market, right? Like you must have heard to it. So sky's the limit. I think as people understand more and more about the biology of these cells and how this treatment can be managed, and then how these cells behave in the patient's body, like in future, like they can be used for various other diseases too. I think I'm pretty sure. And also now the field is also moving into in vivo editing. So instead of using this cell therapy, people are giving these vectors into the human body and try to make patient's own T cells or NK cells or immune cells to express these markers and make them functional. So like we are in the exciting time where this engineering field is booming, and there are a lot of cutting edge technologies are developed, like for example CRISPR and all these technologies. So I think in future sky is the limit. I'm very excited about the future.
Yeah, we were talking about the affordability of these cell therapies. So off the shelf is a very fancy term and also very important term for us. So what do you think about this kind of therapies? Can the poor countries or low and middle income countries can ever get this kind of therapies very soon? Or it's going to be very difficult, even with the off-the-shelf therapies, because beside that, I think there are the problem of the reagents of manufacturing in their domestically. I think it's already a very difficult task. So how can this happen in the near future?
I think that's why off the shelf is very important, especially because of the affordability, right? Not only actually not like we think that everyone in US can afford this therapy, but even in US there are people who cannot afford this CAR T therapy, right? People who goes to these community hospitals, because we talk about this, but we need to understand that even in US, like community hospital cannot give this CAR T therapy to their patients because they don't have expertise or resources to give these therapies. So this off-the-shelf is not only for poor countries, even for rich countries, this affordable off-the-shelf therapy is highly warranted. Okay, and especially for poor countries, One thing, if it's off the shelf, and if we can manage this logistics, we may not have to produce in that particular country. For example, I'm saying in Nepal, if we wanted to make our own off the shelf, even though we are not able to manufacture it, but if we can transport the product either from India or US or some neighboring countries, it's still valuable, right? It's still patient can access to these therapies. Because imagine like even CAR T, like compared to US, India is making it in 10 times less cost, right? In $30,000. This off-the-shelf product will be even significantly lower. So I can tell you, don't quote me on this, but we can make these products like come down to few thousands, right?
Yeah.
Maybe below 10,000. So if we can make this off-the-shelf product, which is really affordable for these patients, I think in future, people in the lower developing countries or in third world countries can also access it. Even if they cannot manufacture in their own countries, they can get it from these other places if they can figure out about these logistics. Because all you need is a freezer and a transportation, right? Like where you can freeze it, and a good medical system where doctors are aware how to infuse this product into these patients. So I see this product will be beneficial for the countries like Nepal or other third world countries.
So one of the recent news was like NIH has prioritized the organoid alternative to the mouse model, I think, organoid research. I mean, the organoid as an alternative to the mouse model. So what do you think about this in the cell therapy thing? Is it like, are you already using this kind of model in your lab? And how much?
Yes, so definitely we use this kind of models in our lab. We use organoids as well as spheroid models. And I think it's a good, I don't know, it might take some time, but it's a very good initiative because, because of the animal welfare, and especially for our field, it's always because, we are working on this cell therapy, which are taking for human cells, and we cannot use, the only mouse model we can use is NSC or immune deficient mouse models, right? So in these immune deficient mouse models, we cannot really understand the interaction between these immune cells, right? That's why actually mouse models are, although we are required, we see this efficacy, but they are not very suitable in order to understand the mechanism. Because when we use this cell therapy into the humans, the outcome is really different compared to the mouse outcome. Actually, it's even better. So that's the advantage of cell therapy when it comes to mouse models and in human. So if we can, we are trying to develop these organoids where there is not only a presence of tumor cells, but also a presence of immune cells and all other cells that are present in immune microenvironment. And then that will recapitulate what's happening in patients, and we can use our cell therapy product in these models to better understand what's happening in human beings. So that's also, we are working on it. And I think that's a good initiative. I don't know how long it will take, but maybe in near future, and there are a lot of companies now, they are strictly working on developing this organoid, which is mimicking the exact tumor microenvironment in the patient. Still, I don't say it will be happening in 1 year or 2 years. Still need some years for a proper research to really develop this system. But definitely the science is moving toward that field. And I won't be surprised in next 10 years, people will strictly use organoid models and stop using these mouse models. So I won't be surprised.
Yeah, we are already in the golden time. We are doing amazing science and also being able to translate. So it's a crucial time, I think, in terms of most of the biological innovations happening around the world, I think. And now I think also not just about the science, also we are caring about the accessibility, affordability, how we can democratize these scientific innovations all around the world. This is also a good part of the science these days, I think. So we are already at the end of our conversation. So at the end, I would like to ask you about some suggestions, that you want to give to the graduate students who are looking for opportunities in this field, like yours. Yeah.
So first thing, the first thing I would say is build this strong scientific foundation. Okay, like don't like as a graduate student, you need to build that strong scientific foundation in order to excel in the future, right? Please go on hands-on, do the experiments, fail on the experiments, because no matter how much you study, until and unless you don't go and do those things and you fail by yourself, you will not learn. Okay, so that would be one of the most important. Go hands-on, do the experiments, fail. Like how I think there was one Addison or someone said, it's not that I failed, I learned 1,000 ways that didn't work, right? So that's very crucial. And then thinking back, I think the world had changed, the science had changed. It's please, if you can, I could not do it, but if I could go back and like go back on time, I would definitely learn some of this like this bioinformatics stuff, where you can learn a little bit about this programming, or how you can understand the data, like have some knowledge about this, because the science is changing so much, right? Science is like fashion, like something is hot now, the things that are hot 10 years ago, that is cold now, and new things will emerge. But somehow the cell therapy has been for past 10-15 years. It's here, and it's here to stay. So learn about some bioinformatics tools, like at least have some basic knowledge on those things. And of course, like collaboration, right? Think about collaborating with your colleagues or seniors. Have some good mentor, talk with them, and whatever you think you are not great at, try to have some mentor who can teach you or give you some advice, or train you how to bypass those weaknesses and then utilize your strength, right? So you cannot learn everything, right?
Yes.
So whatever you learn, be good at something. Like pick one thing and be good at that thing, okay? And also don't get fixated into what is popular or what is fancy now. Think about the future. Think about what's going to happen in next 5 years or 10 years, right? So think about it, and then try to learn about it. So that's what a few things I want to give it tell to graduate students, because nowadays people are smart. It's not like with the advancement of the internet, you can learn whatever you want. You don't have to go to college now, right, to learn about things. So just be hungry to learn, and then always try to learn from your mistakes.
Thank you so much, Dai. Thank you so much for this conversation.